welcome to lecture 4 in our series, and in this lecture we'll focus on prokaryotic organisms. Again, let's put everything in context. So we said that the tree of life consists out of prokaryotic and eukaryotic organisms. The prokaryotic organisms being the archaea and the bacteria, and the eukaryotic organisms being the fungi, the protists, the animals, and the plants. So we've talked a little bit about prokarya and the comparison between eukarya, that prokarya have no nucleus, they have a nucleoid, they have no internal membrane systems the way prokaryotic organisms have it. They have no membrane-bound organelles, they have no interior compartments like eukaryotic cells have. One example for this are the vacuoles and plant cells. So prokaryotic organisms don't have that. They have a basic cytoskeleton and a rigid cell wall, which is often important as they often live in very harsh environments and need to protect the inside of the cell against the outside. To have you get a feeling for the size of the size difference between eukaryotic and prokaryotic organisms, I've incorporated this picture here, which shows here a eukaryotic cell, and this patient here suffers from a bacterial infection, and you can see here a cell of this patient, and it has this very characteristic nucleus, so it's a eukaryotic cell, and these here are the bacteria. So you can see that they are very small compared to eukaryotic cells, and as you can see in this picture here, if this is a bacterial cell, bacteria usually range in size between 1 and 10 micrometers, so they are very small, depending on the species. They have no internal membrane system. They have their circular DNA, which is shown beautifully, illustrated beautifully here in this picture. So it's circular. And their flagellum consists out of a protein that is called flagellin. And this is very different from eukaryotic organisms, and I will compare flagella in bacteria with flagella in eukaryotic organisms, and you will see that there's a huge difference. So although those flagella may look very similar to those in eukaryotic organisms, they are structurally very different. It's also that they move very differently, which I will show you in one of the next slides. The cytoskeleton of the bacteria, they have similar structures like we know them in eukaryotic organisms. Namely, we have actin in eukaryotic organisms, we have the intermediate filaments, and we have the microtubules. The microtubules being the largest ones with about 23 nanometers in diameter, and the actin filaments with about 6 nanometers being the smallest of them. And prokaryotic organisms do have actin-like filaments. In this case, they are called MREB proteins, and they also have tubulin-like cytoskeletal components, which are called the FTSZ proteins. What we will learn is that these proteins, or these cytoskeletal components, are critically important in prokarya for division of the cells and also for metabolic processes. And interestingly, there's another group within the prokaryotic organisms that we have not described in eukaryotic organisms, and these are the MIND-PAR-A proteins. These seem to be characteristic for prokaryotic organisms. The reason why I bring this here is that bacteria and in general prokaryotic organisms are critically important for our ecosystems as they break down organic material and get their energy out of this organic material, and they critically contribute to the homeostasis in our ecosystems. Without bacteria, our ecosystems would not work. However, there are some bacteria that are less desirable, and these are the pathogenic bacteria. And as you may know, antibiotics can be used in order to get rid of these pathogenic bacteria. And what many antibiotics do is that they target the integrity of the cell wall of those bacteria. However, many organisms or many bacteria are getting immune to those antibiotics. So the antibiotics, in other words, do not interfere with the integrity of their bacterial cell wall anymore, and for that reason those bacteria are not harmed by the antibiotics, and the antibiotics are not effective. What newer antibiotics are now targeting are these cytoskeletal components within the organisms. And since those cytoskeletal components are critically important for things like binary fission, for cell division within those single-celled organisms, this is a very important part of research. Also, with regard to how do bacteria get their energy, so we've talked a little bit about the cyanobacteria, that the cyanobacteria have a cell membrane in them, and within the cell membrane there's a protein machinery that is able to perform photosynthesis. And the byproduct of this photosynthesis is oxygen, and the bacteria get their energy out of this photosynthesis, and the energy-rich bonds they can synthesize by themselves out of this photosynthesis. Other bacteria break down organic materials and are for that reason critically important for our ecosystems. So the cell wall of bacteria, there are two main classes of bacteria, and you might have come across these words. These are gram-positive and gram-negative bacteria. And they're called gram-positive or gram-negative if they can be stained for a certain stain that was developed by Hans Christian Gram, who lived from 1850 to 1938. And what he discovered more or less by accident was that some bacteria take on this stain, whereas other bacteria cannot be stained by this dye. And this has turned out to be critically important in figuring out what kind of bacterial infection, for example, a patient has, and what kind of antibiotics will be useful for this infection. And the background to this is in the differences in cell wall of those gram-positive and gram-negative bacteria. So if we start here with the gram-positive bacteria, you can see that they have a cell membrane here. This consists out of lipids and proteins, these phospholipids and proteins. But they also have an outer cell wall, and this outer cell wall consists mainly out of peptidoglycan. Whereas the gram-negative bacteria, they also have their cell membrane. They also have peptidoglycan, but this peptidoglycan cell wall is much thinner and incorporated in the middle here of the cell wall. So it's only one part of the cell wall. The other part of this cell wall here consists out of lipids and carbohydrates and proteins, and is on the outside. And for that reason, because the cell wall of these bacteria is different from the cell wall of these bacteria, these ones do not take on this dye that was developed by Hans Christian Gram, and for that reason are gram-negative. And now if you think about this, so the antibiotics that we are using in order to fight bacterial infections often interfere with the integrity of those proteins and carbohydrates within the cell wall. So with other words, the cell wall destabilizes and the bacterium is no longer protected against the outside environment, and for that reason disintegrates. In order to know which antibiotics will be effective, we need to know what kind of cell wall these bacteria have. And as the Gram stain allows us to distinguish those two types of cell wall, this is often used in order to identify what kind of treatment the patient will receive. Now, if we talk a little bit more about the flagella and the differences between prokaryotic and eukaryotic organisms here. Flagella are used for these single-celled organisms in order to move within limits and come from point A to point B. And what you will find in bacteria is that these flagella that are formed out of flagellin protein, they rotate, very much like the motor of a motorboat, they rotate. And this is driven by a hydrogen gradient, and we'll talk more about how hydrogen gradients will make structures rotate when we talk about the electron transport chain during metabolism. So what you will find is that these flagella are incorporated into the cell wall and cell membrane. So if this here is a cell wall and this is a cell membrane, then those flagella are incorporated into this, and they basically rotate. And for that reason, move the bacterium forward. In eukaryotic organisms, those flagella don't rotate. Those flagella undulate from side to side because the mechanism at which these flagella move is critically different from these prokaryotic organisms to the eukaryotic organisms. Here we have a hydrogen gradient that causes the rotation of this flagellum, and for that reason we have a rotating flagellum. The diameter of those flagella is only about 20 nanometers. The flagella in eukaryotic organisms are much bigger. And they have no cell membrane that surrounds those flagella, unlike eukaryotic organisms. So how do bacteria and in general prokaryotic organisms divide? So they divide by a binary fission. And what that means is you have one mother cell, and within this mother cell, the circular genome is duplicated, is then divided within the mother cell. So you have one copy on one side, you have the other copy on the other side, and then the cell elongates and it will cleave in the middle, and you have two daughter cells. And this is named binary fission. So you have one mother cell, genome is duplicated, and then the cell elongates, it is cleaved off in the middle, and you have two identical daughter cells. And critically important structures such as ribosomes will be distributed between both daughter cells. So here, if you have your genome, you have the origin of replication, and in this case here on the side, the genome replicates, then one copy will be arranged on one side of the cell, another copy will be arranged on the other side of the cell, the genome divides, the cell divides, and then we have two individual cells. And it is the cytoskeletal, these are cytoskeletal components that play a critically important role in this division, in transporting proteins and molecules from one side of the cell to the other side of the cell, and also in this pinching off, in this cleavage, and in the elongation of those cells. So cytoskeleton is basically like the interior skeleton of the cell that enables the cell to perform these functions. So this is all that I have to cover on prokaryotic organisms, and in the next lecture we'll talk more about eukaryotic organisms.